Hello, everyone. Thank you for taking the time to join the Context Therapeutics webinar today. The discussion is going to be recorded, so it can be accessed at a later time for those who cannot attend. Also, we will be hosting a Q&A session after the presentation. If you have any questions, please submit them through the Q&A function at the bottom of your Zoom window. On Tuesday of this week, Context Therapeutics announced the selection of CTIM-76, a T-cell engaging biospecific antibody, as its lead clinical development candidate to target CLDN6 positive cancers resulting from its research collaboration and licensing agreement with Integral Molecule. We have Context management here and two experts with us to talk through the program and the selection process. Martin Lear is the CEO of Context Therapeutics. We also have CFO Jennifer Manai on the call to help answer questions as needed. Marty, maybe you can take a minute to introduce yourself and then we'll pass it to Eric and Joe.
Thanks, Sue, and it's a pleasure speaking today, and thanks to everyone who's joining us for this webinar. It's certainly an exciting time for Context Therapeutics and the announcement of our clinical candidate for CLDN6 positive tumors in CTIM-76. Marty Lear, co-founder and CEO of Context Therapeutics. It's a pleasure to be with everyone today. Turn it over to Jen now.
Hi, everybody. Jennifer Manai, CFO, Context. I'll hand it over to Eric.
Eric Butz, acting as scientific lead for this project. I have a background in cellular immunology with 16 years at Immunex and Amgen, and then helped found a private consultancy, which I've been doing for the last 7 years.
And hi, I'm Joe Rucker. I am the research lead for the CTIM-76 project. I am the Vice President of Research and Development at Integral Molecular. I'm also a co-founder of the company, and we're happy to be working with Context Therapeutics, moving this program forward.
Thanks, everyone. So I advise everyone to take a minute and review our forward-looking statements and important notices and disclaimers. We may be making forward-looking statements throughout this presentation, and we will be referencing historical preclinical and clinical data throughout the presentation with various footnotes to call that out. From a process standpoint today, I'll give a brief overview on Context Therapeutics. That will be followed by Eric, who will delve into CLDN6, the history of the target, why we believe it's a fascinating and relevant target, some of the advances that are going on in the field. And then we'll progress into Joe's presentation on the discovery of CTIM-76. Integral, and then more recently, Context, our partnership, has spent a long time, in this case, even years, on the target. For the purpose of today's conversation, we will just focus on CTIM-76. We'll leave future presentations for how we evaluated a much broader array of bispecifics, what that comparative data looks like, as well as additional data at subsequent presentations over the course of 2023 at various academic conferences. But today, just focused on what we believe to be our new exciting asset, CTIM-76. And then at the end, I'll provide concluding remarks, focus a little bit on competition in the Claudin space, where those programs lay from a development standpoint, and then we'll open it up to the group for Q&A. You'll notice, I think most people are pretty familiar with Zoom at this point, but at the bottom of your screen, there is a Q&A button. If you have a question, feel free to submit the question in that section. We will note those questions and do our best to address them as time permits at the end of the presentation. So with that, for those of you who are unfamiliar with Context Therapeutics or need a little update, Context is a publicly traded company. We're traded on the Nasdaq, and we are focused on women's oncology, particularly cancers of the female reproductive organs, so breast, ovarian, endometrial. We have two programs that we're developing. The first I'll speak about is ONA-XR. That's our oral progesterone receptor antagonist that is in a number of phase 2 clinical trials, as well as a phase 1b/2 trial in collaboration with the Menarini Group. We announced 3 weeks ago positive preliminary data in endometrial cancer, where we showed that 77% of evaluable patients in that data cut met the 4-month PFS target, which is one of the primary endpoints for that trial. So we're really excited about the prospect of ONA-XR in endometrial trial. And then we'll be actually providing an update in breast cancer next week at the San Antonio Breast Cancer Symposium. So a lot of activity going on in that program. And then today, the subject matter is CTIM-76, our CLDN6 program. That program is progressing into and through IND-enabling studies with an eye towards filing an IND in the first quarter of 2024. Contents went public about a year ago. In the fourth quarter of 2021, we raised across 2 financing gross proceeds of $60 million. We have capital into Q1 of 2024. So we have cash for 2023 and a little bit beyond to achieve a number of significant milestones that are further described on slide 6. We have 4 ongoing clinical trials. The first of which that is described here is our partnership with the Menarini Group. The trial is referred to as the ONA trial. That trial initiated this quarter, in the fourth quarter of 2022. And then the next milestone for that trial will be phase 1b data at the end of 2023. The second trial here is a combination trial for ONA-XR with fulvestrant. That trial will present preliminary data next week at the San Antonio Breast Cancer Symposium. And then just below, we present updates on our 2 gynecologic cancer trials. I mentioned the exciting data from a couple of weeks ago in endometrial cancer. That trial is ongoing. We believe the next clinical update will occur in mid-2023. And then our trial in recurrent granulosa cell tumors of the ovary continues to progress. Stage 1 of that trial has completed enrollment, and we look forward to providing a data update for that trial in mid-2023. And then certainly CTIM-76, we'll spend a lot of time today talking about the achievement of the nomination of a clinical candidate, and then moving beyond that, the IND filing in Q1 of 2024. And I will reference, even though this slide may be hard for people to see, the slide deck is fully available on the SEC website for download. So if you're having any trouble viewing this, I would encourage you to download the slide deck. And so with that, I'm going to turn over the presentation to my colleague, Eric, to discuss CLDN6 target biology.
So as Marty's mentioned, the target of CTIM-76 is CLDN6. It's a tumor specific protein that can be expressed at high levels across a number of different tumors. It's expressed at very low levels or absent from normal adult tissues. It's conformationally dependent antigen, so it's challenging to target with antibodies. And what makes it more challenging is that its external sequences are highly conserved and very similar to Claudins 3, 4, and 9 in particular. Selectivity is very important for this project to avoid off-target toxicities because of its expression on a number of other tissues. And there's clinical validation of the target with BioNTech's BTN-211 CAR T cell, which has published phase 1 data with solid tumors, and they've had really good response rates in some of their cohorts. The unmet need here is that by targeting Claudin-6, you could target a number of cancers which are poorly addressed at the moment, including ovarian, testicular, small cell lung carcinoma, a couple of others, but that it's a difficult target to hit because of its similarity to other claudins. CTIM-76 has excellent potency and given its selectivity over other targets, good safety, and the format is easily manufacturable. Background on the claudin protein family, it's a tight junction protein. Tight junction proteins are important for maintaining boundaries and tight associations between cells at surfaces to prevent to maintain barrier functions. 27 different claudins have been characterized to date, and there's dysregulation of tight junction proteins in various forms of disease. With claudins in particular in oncology, they've been shown to contribute to issues including metastasis and invasiveness. There are a number of proteins in the family, as we've already mentioned. Each of them is, or many of them are associated with different kinds of cancers. As you can see here, claudin 6 is of particular interest given that it's expressed across many different kinds of cancers, especially as cells dedifferentiate, where as they assume less differentiated status, they can upregulate claudin 6. You can see the list here, and the biology is consistent with the interest in the family. Claudin 6 is particularly closely related to claudin 9, but also to claudins 3 and 4. The relatedness tree is here on the left. Because also the extracellular portions of the molecule are quite small, that makes targeting difficult. And the claudins 3, 4, and 9 are expressed on a number of different tissues like pancreas, kidney, and within the ear. So selectivity is really important to avoid off-target toxicity effects. These are the structures of the claudins. The two extracellular portions are there above the cytoplasmic membrane. In ontogeny of tissues, claudin 6 is expressed and helps define the boundaries for things like the tissues of the lung. And then as the tissues mature, claudin 6 expression is downregulated so that in the adult tissues, it isn't expressed under normal circumstances. As cells become cancerous, however, that control is lost and claudin 6 can become upregulated. Therefore, it becomes a good target for us to look at in that context. As an example of its expression in normal versus tumor tissues, on the left, you can see a large panel of normal tissues without expression of claudin 6. And then on the right, you can see examples of testicular, ovarian, and lung cancers and their high expression of the target. Not only can it be highly expressed, it's also consistently expressed across the tumors. So that makes targeting the tumors much easier. You can see here a panel of different sites from testicular cancer and ovarian cancer with broad expression of claudin 6 across the tumors. That makes things like escape variants more difficult, and it makes it easier to address the entirety of the tumor with the therapy. It has the potential to reach a large number of patients in addition to targeting a number of different tumors. So here you can see a selection of different tumors where claudin 6 has been defined to be expressed, including the 4 main ones at the top, with incidence, rate of relapse, and the rate of claudin positivity in those groups, and the patient populations on the far right-hand side based on the rate of relapse to disease. So particularly with lung cancer and ovarian cancer, there are large groups, but also with potential for treatment of breast and endometrial cancers too. Here's a timeline for research developments with CLDN6. It was defined as a target in a Chugai patent in 2009. In 2016, Astellas acquired Ganymede, partly because of interest in the IMAB-027, which targeted CLDN6. In 2019, IMAB-027 failed to show good activity in phase 2 because of lack of activity. We'll get to that in a minute. In 2020, AbbVie showed poor tolerance in a phase 1, probably because of lack of selectivity. In 2021, Xencor and Novarock also announced programs. And then in April 2022, BioNTech published some clinical data showing that their BNT211 did show some efficacy in a phase 1 study, which we'll show a little bit of. And then IMAB and Amgen have also announced programs in this space. The different strategies have been addressed by the different groups. So the original antibody that failed in the clinic was a monoclonal antibody designed to target CLDN6 and block its activity. Ours is a bispecific antibody designed to engage T cells. The BioNTech molecule is a CAR T cell, and antibody drug conjugates require receptor internalization. For our purposes, for CLDN6, the antibodies alone have showed that CLDN6 activity is not really dependent on interfering with the signal. The bispecific antibody has shown proof of concept with a BioNTech bispecific. They're not pursuing that though. They're looking at their CAR T cell. ADCs haven't worked well in this space because of poor internalization. We have internal data showing that you need very high concentrations of an ADC to make that work. And the CAR T cell has clinical proof of concept. So the IMAB-027 data that showed poor efficacy was with the antibody. You can see on the left some of their data and the reasons why they didn't proceed. They had only very weak responses with the antibody. On the right, you see our data with an ADC showing that you need very high concentrations, much higher than you'd achieve in vivo to get killing with that. The BioNTech BNT211 is an interesting approach. It's a CAR T cell targeting CLDN6. And then because CAR T cells can have shown not to persist very well or to have optimal activity over time, they've also included as a co-therapy an mRNA vaccine encoding CLDN6. And their goal was to use that to achieve optimal levels of activation of the CAR T cell after transfer by treating with the CARVac periodically after transfer of the CAR T cell. So the schematic for that was on the right-hand side, where if you use high dose CAR T without the vaccination, you get high levels that then go away, and that maybe you could maintain the optimal levels of activity and CAR T expansion using the CARVac strategy. They've shown preliminary clinical data, which actually looks really good. So if you look at testicular cancer in the left-hand panel and compare that to current standard of care therapies, you can see that they got response rates of 31% with their initial dose plus or their secondary dose of up to 10 to the 8 cells, compared with 13% for sunitinib or 11% for brentuximab vedotin, and no response with pembro in that context. In ovarian cancers, they had 50% response rates with the BTN-211 plus the CARVac, as opposed to 32, 28, or 27% for the standard of care therapies with mirvetuximab or pembro plus nemvaleukin or bevacizumab. The limitations of the CAR-T plus CARVac approach are that it isn't especially dose sparing for the CAR-T. In the phase 1, they've used up to 10 to the 9 cells. What they use for a phase 2 dose has yet to be determined. And that's really similar or in excess of what's being recommended for other kinds of CAR-T cells directed at claudins. The CAR-T have limited durability, or the response has limited durability, so that you can see that where the tumor wasn't entirely eliminated, the tumors begin to grow back again in the middle panel. And the CAR-T have limited persistence. And the data on the right-hand side there aren't exactly T-cell counts. They're from flow cytometry. You can see that after transfer, even with the CARVac treatment, that the cells decline over time. And then you have the potential for toxicity effects too with the CARVac, because you're expressing Claudin-6 in tissues where it might not normally be expressed, and that results in activation of the CAR T cell, but may also result in some toxicities. So, for example, with the BNT211 plus the CARVac, there were some signals of liver toxicity with transaminases and lipases being elevated. One of the places where the CARVac gets expressed is in the liver, so that may be related to that. It's also a complex therapy, being a CAR T cell therapy, given that you have to make the CAR T using cells from the patient, as opposed to CTIM-76, which is an outpatient infusion of an off-the-shelf reagent. The CAR T cell treatment requires lymphodepletion as opposed to the bispecific antibody, and can be repeated as necessary without having to either add more CAR T cells or expand them over time, where they still seem to peter out. So that's the biological background. And now for Joe Rucker will describe the discovery and development of the CTIM-76 molecule itself.
Great, thank you, Eric. So obviously, Claudin-6 is, given, what Eric was just describing, Claudin-6 is a very exciting target. And when we sat down with Context Therapeutics to think about what would make an optimal bispecific molecule against Claudin-6, we had a number of considerations. So on the Claudin-6 side, we wanted, of course, high affinity tumor antigen binding to Claudin-6. But of course, the specificity was really getting that right was paramount, particularly against the widely expressed claudins and closely related claudins, Claudin-3, Claudin-4, and Claudin-9. We wanted to do a T-cell engager, and we decided on going after CD3. And when choosing a CD3 arm, we wanted an arm that was clinically validated, one that we had freedom to operate. And then we wanted to explore a range of potencies and epitopes, really to find what was going to be the best arm for us to work with. In terms of the geometry of the bispecifics, we took an agnostic approach. We wanted to try a number of different formats that differed in geometry, in valency, and then exploring, of course, the different types of Claudin-6 and CD3 arms that we had access to. And then, of course, more generally, thinking towards the clinic, we wanted these molecules to be cross-reactive with non-human primates, so we could, have more straightforward IND-enabling studies. We, of course, would like Optimally, we wanted a molecule with an FC so we could have a good half-life. And then, of course, silencing variants in any FC to eliminate any potentially adverse effect causing effector function. Oops. So let me introduce everybody to CTIM-76, which is our development candidate. So as you can see, it's got a single Claudin-6 binding arm and a single CD3 binding arm. The Claudin-6 binding arm is high affinity and very selective for Claudin-6 over the other Claudins. The CD3 binding arm, it's functionally monovalent and has really been chosen to avoid aberrant T cell activation. So we have a good safety profile, and we're using a silenced FC domain to avoid the T cell activation. As you'll see from the following data, we have really strong T cell dependent cell cytotoxicity. And we're able to get that cytotoxicity with no or minimal cytokine release. And all of this antibody is a humanized antibody, as to be expected, to really lower the immunogenicity risk. And a lot of the early developability and manufacturing work has really shown that it's a very stable molecule and providing very good yields. So let me tell you how we got here, where this came from. So, the Claudin-6 arm originally came out of Integral Molecular, and we went after this problem. We looked at this and we said, okay, Claudin-6 is very, very highly conserved. It's highly conserved with Claudin-3, Claudin-4, and then Claudin-9 was very high conservation. In fact, there are only 3 amino acids difference in the extracellular regions between Claudin-6 and Claudin-9. And as you look at the structure, you realize that only one of those residues is accessible and something that you could use to get antibody specificity. It's a complex membrane protein, and it's the antibodies, the antigen is very conformation dependent. So how you go after the antibody is going to then develop the antibody is going to make a big difference. And because of that high sequence homology, it's got this very high sequence homology with mouse, we needed to use a divergent species for immunization. And I should say we originally used the chicken for immunization, although the antibodies and the data we're showing you is going to be on a fully humanized molecule. So when we went after this, we have developed at Integral these very specialized techniques for going after these challenging complex membrane targets. And we isolated these antibodies with the picomolar affinity and very high specificity for Claudin-6. And in a work that's in a manuscript that's in press, we actually did a lot of characterization to see how we could get such precise specificity of these antibodies, and we're able to show that how an antibody can really recognize a difference at a single amino acid, really at almost atomic level type of resolution. So when we began to look at these at the antibodies, at the parental antibodies, the parental IgGs at Integral Molecular, we wanted to really make sure that they were highly selective for Claudin-6. We compared all of our molecules to the IMAB-27, which Eric discussed as a benchmark in the previous section. And you can see, for example, on the left is sort of the selectivity studies that we performed on IMAB-27. It's got strong Claudin-6 binding, but you can also see a very strong binding to Claudin-9 in blue. And so really there are some selectivity, there's definitely improvements in the selectivity that one could go after. And if you look on the right, IM301 is the parental for CTIM-76. And you can see that it's very, very selective for Claudin-6 in red over the other 3 Claudins, in particular Claudin-9. If you look at CTIM-76, in its particular format, with its monovalent arms, you see very sort of excellent specificity with strong binding to Claudin-6, but essentially negligible or no binding to any of the other claudins. And these binding studies were done with highly overexpressed targets. So this is an exceedingly specific antibody. But we wanted to look at specificity beyond the claudins. And we used at Integral Molecular, we have what we refer to as our membrane proteome array, which is 6,000 human membrane proteins that are individually overexpressed in living cells in their native state. And we could look at the broad cross-reactivity of this antibody across the entire human membrane proteome. And our thinking is that an antibody that has the correct specificity is really going to have fewer adverse effects. So this is work that was done on the parental antibody. This is the CTIM-76 specificity work is essentially in process. But what we saw is that we saw good binding to Claudin-6, no binding to Claudin-9, some low binding to Claudin-4. This was in a sort of a native IgG1 without effector function silencing. So we saw Fc gamma. And then interestingly, we saw an off-target binding to another protein, ABCC3, but extensive work at Integral showed that this antibody was against an intracellular epitope and would have no effect on influence on CTIM-76 as a therapeutic. So let's go into some of the more sort of the key details about CTIM-76 as a bispecific. So we, of course, wanted to look at lysis, T-cell mediated lysis across different cell lines. On the left is the ability of the CTIM-76 to lead to T-cell mediated cell killing in overexpressing Claudin-6 line. And you can see that it has essentially sub-picomolar EC50s, so very, very potent. Looking at a ovarian cancer cell line, OV90s, we also see picomolar EC50 and cytotoxicity. But in a negative cell line, an HEK cell line, we see very, very low levels of some non-specific killing at the nanomolar range, and it's very low. It's very, very low levels. So pretty much as you go to high, medium to low levels, you can at the levels of Claudin-6 overexpression, you see very potent killing, and then in negative controls, you see negligible. This is, of course, important to look at in terms of cytotoxicity across the different claudins. On the left, you can see that in terms of Claudin-9, we have a 3 order of magnitude selectivity in terms of cell lysis. The cell lysis versus Claudin-9. In the center, it's close to similar with Claudin-3 and also similar with Claudin-4 on the right. So the selectivity that we see in binding is really converted over to the selectivity that we see in terms of cell lysis versus the other claudins. And that's roughly 3 orders of magnitude of selectivity gives us a sort of a wide range that we can potentially dose our molecule. In terms of the cytokine release, which of course is really what determines the therapeutic window for these molecules, CTIM-76 has a wide therapeutic window. What we did is we looking at cytokine production in the overexpressing Claudin-6 cell line model. What you see is for a variety of different cytokines, interferon gamma, IL-2, IL-10, TNF-alpha, you see a wide, roughly 2 order of magnitude window between the EC50 for cell cytotoxicity and the EC50 for cytokine release. And what this means is that at levels that this supports the dosing of CTIM-76 at ranges where we'll get good cancer cell killing, but have manageable levels of cytokine production. We, of course, looked at this not only in the overexpressing line, but in the ovarian cancer line, OV90, and we see very, very similar results where there's a good gap between the EC50 for cell lysis and cytokine release. Once again across the same set of cytokines. So I just want to end, as Marty alluded to, we would at future academic meetings, we're going to talk in more detail about how we went through the selection process for CTIM-76. But I want to just sort of end with this, which is that as we started this program, as I mentioned, we looked at a variety of different formats. And this looking at broad levels of formats really enabled us to pick a format that had the optimal properties we're looking for. As an example, what you see here is a comparison of CTIM-76 with the exact same arms in the format of a tandem scFv, also known as a BITE format. And what you can see is a good, in this case, 28-fold gap in the activity of CTIM-76 versus the lower activity of the BITE molecule, and really suggest that this approach that we took for selection has really given us the optimal candidate. And we look forward to presenting more data about CTIM-76 and our selection process at future meetings. And at this point, I'll hand the floor back over to Marty.
Thanks, Joan. A great overview on CTIM-76, and that followed Eric's discussion about the potential of Claudin-6 as a whole, knowing that it's expressed in a wide variety of tumors, that expression is both at high density and remarkably consistent and homogeneous across those tumors, ideal properties for a tumor-associated antigen that we want to take advantage of with our bispecific format. So with that, I thought it'd be helpful to frame the competitive landscape for Claudin-6, not just our program within Claudin-6, which we'll talk about, but Claudin-6 as a whole. So oncology is an area that many pharmaceutical companies and emerging companies are spending a lot of resources trying to develop technologies and platforms and exploiting various targets that are out there. And so we're always looking for targets that are as validated as possible, where there's minimal competition and addresses a large patient population, sort of the unicorn target. And so what we did is try to frame as many what we would call interesting targets as possible. Obviously, there's more targets than we could ever list on this slide, but we picked the ones that seem to come up the most in discussions with clinicians and investors. And we frame them on 2 axes. So looking at the target, whether it is validated, the most validation being FDA approval in the case of CD19 and CD20 and BCMA, or emerging, where it's really very, very nascent and potentially no clinical proof of concept, and then large and small population. And so again, going back to the disclaimers at the front of the presentation, knowing that this is a subjective exercise, what we thought would be helpful is if you look at the top right quadrant, there's a handful of really interesting targets. And not surprising to us, 2 of them are Claudins, Claudin 18.2 and 6. But if you look at 6, what is unique about it is it addresses a surprisingly large population. So Eric talked about earlier that we, based on our analysis of the relapse remitting population in the United States, that's biomarker enriched based upon published data and our own data, we think there's about 62,000 patients in the United States alone. That's a large potential market for Claudin-6 targeted therapies. And from a validation standpoint, we now know that BioNTech has treated 21 patients. The data, particularly in the dose level 2, is showing really promising efficacy. So in DL2, they had a 50% response rate. And so for us, that's the validation we needed clinically. And so this is a target that I think stands on its own and is in good company with other targets of interest like PSMA and DLL3 and PRAME, which had some really nice data at ESMO this fall. As you look at competition within Claudin-6, the way I would view it is it's very much emerging. BioNTech, as I mentioned, has treated 21 patients with their BNT211 program. But the other phase 1 programs have literally just entered the clinic. Those would be Daiichi, a second program from BioNTech, which is a bispecific antibody, but that is mRNA encoded. And then out of China, there's a CAR-NK approach with substantial gene editing associated with it. And so there's 5 programs in the clinic conceptually. Amgen is actually has entered the clinic, but is not enrolled or opened their trial formally. So we think it's still very early days. And we look forward to seeing how some of those programs progress to confirm the BioNTech efficacy signal and then enable us, hopefully, to differentiate from a pharmacokinetic standpoint with our IgG background and the potential for a 2 to 4 week dosing paradigm, as well as a potentially wide therapeutic window relative to some of these other therapeutics. On the preclinical side, it's always harder to gauge competition because earlier stage, less information out there. There are 2 additional ADC programs, one out of China, that's a dual Claudin-6, Claudin-9 targeting antibody. We understand from the Stemcentrx and AbbVie experience with SC004 that off-target binding to Claudin-9 carried side effects associated with hearing loss and GI barrier instability. We'll wait to see if that's confirmed from this Chinese company. And then UCLA, through the partnership with Toral, has a program as well. As Eric mentioned earlier in the presentation, Claudin-6 is an integral membrane protein. Those proteins historically do not internalize. That would actually be very bad from a cell barrier standpoint. And in our own hands, we don't see it internalize. So these therapies could absolutely work. That would most likely be achieved based upon our analysis through bystander effect. And typically when you're using a bystander effect with an ADC, there's substantial toxicities associated with it. So we'll continue to keep an eye on those programs. On the bispecific side, competition is limited. Moving from left to right, there's an emerging Chinese company called Novarock that is targeting Claudin-6, but they're using 5-4-IBB as the T-cell engaging arm. That's followed by Xencor with an undisclosed program, our program, and then a 2x2 from IMAB, also targeting 4-IBB. If you look at those programs in a more granular sense, moving from top to bottom, BioNTech is in the lead currently with BNT211. That's their Claudin-6 CAR-T plus CARVac. They have been updating the clinical community over the last 18 or so months on that program. The most recent update was at ESMO this fall. That data is incorporated in this presentation and was presented by Eric. That program also recently received PRIME designation in Europe for testicular cancer. As I mentioned, BioNTech has a second program, BNT142. That is a bispecific encoding mRNA. it's hard to know why they're doing it. They are experts in mRNA, but in our hands trying to manufacture their program, we had some trouble manufacturing, generating appropriate yields. And so this may have been an end around to some of the manufacturing issues we at least saw. Don't know for sure, but that's another program that we'll keep our eye on. And that just entered the clinic. I mentioned Amgen 794, which is their Claudin-6 BiTE. That program was announced in April at AACR. It is on clinicaltrials.gov, but is not yet recruiting. There's an undisclosed program from China that I mentioned on the previous slide that has extensive gene editing. It'll be really interesting to see. It's also the only natural killer cell approach on here. And so that program is actively enrolling patients. And then more recently, Daiichi disclosed a program and was made available on clinicaltrials.gov using their common chemotherapy that they're attaching to ADCs in the form of DS-9606A. And then you have the IMAB and Xencor programs. IMAB has indicated they will file their IND sometime towards the end of next year, 2023. Xencor announced their program in April of 2021 at AACR. They had a poster, but there have been no subsequent updates or any filings on clinicaltrials.gov to indicate that that program has moved forward. And then historically, as Joe and Eric mentioned, there were two deprioritized programs. The first from Astellas, Gainamed. Astellas acquired Gainamed. Interesting enough, the founders of Gainamed are the same founders as BioNTech. So BioNTech clearly has been working directly or indirectly on Claudins for the last 15 or so years. That program, though, IMAB 027, had lackluster activity, indicating that a MAB may not be the best approach here. We do not think that Claudin-6 in cancer cells has a particularly strong signaling activity associated with it, and so a MAB may not be relevant. And then, as Eric mentioned earlier, AbbVie acquired Stemcentrx, who is developing SE004. That was a ADC with equal affinity for Claudin-6 and 9, and it never got out of phase 1 due to drug-associated side effects. So as people may have seen, bispecifics have become a fascinating space. It was a challenging area until recently for small companies to develop products. And Genentech had their knob and hole patent, which really limited manufacturing of these types of assets. And so historically, companies had to build their own platforms, which was quite an expensive endeavor. With the expiration of the knob and hole patent, that opened the door to smaller companies who may have an advantage developing a particular bispecific towards a target to enter the space. So what we've seen in 2020 is significant pharmaceutical interest in bispecifics. Sister protein Claudin 18.2, which is enriched in gastric cancers, has been the subject of a number of business development transactions over the last year. But we've also seen similar transactions with CD19, CD123, and EGFR. And so I think what's fascinating is, well, certainly CD19 is historically a liquid tumor target. You're seeing an increased interest in deal capacity for bispecific deals in the solid tumor space. To bring it all home before we go into Q&A, we have a product in CTIM-76 that's displaying really encouraging efficacy signals that has a candidate profile, frankly, consistent with what we had hoped for, maybe even beyond at the start of this project. It is, as our colleague, Dr. Evan Dick likes to say, Claudin-6 is a righteous target. And I think we've built an asset in CTIM-76 that is appropriately designed, structured, formulated to target this exciting protein. The drug, through our initial studies, looks quite promising from a tolerance standpoint. So Joe expounded upon nicely the activity gap between cytotoxicity and the activation of free cytokines. That is a really nice component of our drug and something that we hope you all picked up on today. The PK we expect, and certainly through preliminary animal studies, to be consistent with an IgG backbone. And in those same studies, there's been no significant safety findings to date. Obviously, in future presentations, along with the identification process of CTIM-76, we'll present those in vivo data as well. And then we're on track for IND submission in Q1 2024. And we've selected Lonza as our GMP manufacturing partner for the program, and we're very excited to work with them. So with that, want to thank everyone for all of their contributions to the slide deck and the development of this program. I'll open it up to questions now, and our colleague Jen Manai, our CFO, will join as well. I will moderate the questions. I'll be the air traffic controller. And for Joe, and maybe I'll start with you. So one of the first questions we got, Claudin 18.2 and Claudin 6. Claudin 18.2 is really well known, a lot of activity in that space. You saw a couple of slides ago, 4 BD transactions for 18.2 over the last year. What's the difference between 18.2 and 6, and why should someone care about 6?
Yeah, I mean, it's a good question. I mean, from a discovery point of view, Claudin 18.2 is an easier target to go after, but it is a really intriguing target. Claudin 18.2, it's found actually on some gastric cells already, and so it's one of the reasons why people are seeing it as a tumor antigen in a number of gastroesophageal cancers. So that's been a strong unmet need, and it's a target which is relatively straightforward to go after and develop antibodies against. In terms of breadth, I mean, it's been seen obviously in gastroesophageal cancers, overexpression in pancreatic cancers. That's been the primary interest. It seems to be found in a few other cancers, but it seems to be a little bit more limited in its overexpression. What was exciting for us about Claudin 6, and I think is where the interest is that, as Eric showed and you've mentioned, it's found on such a broad series of tumor types just because it gets upregulated. It's not found on healthy tissue, but gets upregulated in the sort of the cancer-associated dedifferentiation. But because of the specificity challenges, it's been harder to go after. And oftentimes when you make antibodies against it, you'll find antibodies that are cross-reactive with 9 or with 4 or with 3. And as we know, that's where the adverse effects come in. So it's broader, but it's been harder to go after just from a discovery perspective.
Very helpful answer. And so one of the questions we got, and this is I think in many cases borderlines on existential. You're either in the CAR T camp or the bispecific. And there's those who say bispecifics, there's no way they'll ever work in solid tumors, and CAR T will never work as well. So how do you think, kind of conceptually, a bispecific versus a CAR T, and what gives you confidence more specifically in CTIM-76 that you will see CAR T-like activity or close to that, with a benefit of, an outpatient procedure, a dosing paradigm that is very different, a cost structure to payers that is vastly different? How do you think about all that?
Who's that one for, Marty?
Both. I'll let you kick it off, Eric, because I can see you're sort of grinning.
I think the clinical response remains to be seen, of course, so that needs to be studied. But the complexity of a CAR T is way beyond the complexity of delivering a bispecific antibody. So that makes it the bispecific an attractive alternative. And we have seen that bispecific antibodies do work in the clinic. So I think it's a promising approach. And the CAR T don't always work in the clinic.
Joe, any comments or?
Yeah, I mean, right now, of course, with, the issue with CARs and solid tumors, of course, has been getting a good persistent response. And as we saw from the BioNTech studies, they're using their mRNA delivery system as a way of sort of getting this, essentially generating the response by creating antigen in other tissues. So I think, at least in the current CAR regime, there's a much more of a potential for off tissue targeting, depending on where the mRNA that gets delivered ends up going. So from that sort of head-to-head perspective, the bispecific is going to be a safer approach.
Yeah, and along some similar lines, if you have any off-target activity with a CAR T, that can be bad clinically, but also really hard to turn off. With the bispecific, it's a much more titratable activity. So if you start to have issues, you can titer back on the delivery of the bispecific.
And there was a question that came in. So to go along the lines of competition, Joe, you showed a little bit of data around what we're calling the Amgen BiTE, the Claudin-6 sequences that we developed with CTIM-76 in a BiTE format. How do you think about CTIM-76 versus a BiTE? What are some of the challenges and benefits of a BiTE versus what we're doing?
Yeah, the always the issue with the BiTEs versus our molecule, there's differences in PK. So a BiTE molecule is a small molecule, gets cleared from the system very quickly, and BiTEs, traditionally have been, without half-life extension, have, required long, 48, 72-hour consistent continuous infusions. CTIM-76, with its IgG-like structure, of course, is going to expect it to have a typical antibody PK on the order of, half-life on the order of days to weeks. So, I think oftentimes getting that sort of long, sort of consistent half-life can give you a better activity. It gives the T cells sufficient time to activate and to kill the tumor. The one question that's still an unknown in solid tumors is how well do the different molecules get into solid tumors. That's a case where a BiTE could have an advantage, but there's really no clinical data to say one way or another. So from our perspective, it's both the extended PK of CTIM-76, and also, as we show, just from a geometry perspective, there's something about the geometry of the IgG format, CTIM-76 format, which gives us an improvement in potency.
That's very helpful. And so one of the questions that came in relates to combination treatments. Is this something that you think we're going to pursue, other companies? What's your view of Claudin-6 combinations?
I think it's a possibility that it could be combined with a checkpoint inhibitor, for example, to see if that improved activity. But the first step for us would be to look at activity as a monotherapy.
Joe, it looks like you had something to say, or that.
Yeah, I mean, it's really sort of along the same lines. As we know that solid tumors can have these immunosuppressive microenvironments. So treatment with checkpoint inhibitors or other inhibitors or other molecules that are in development that help sort of tweak and get the immune system a little bit more jazzed up, I think it's something worth thinking about. But of course, you're going to start with a monotherapy and then use that data to help guide us in future studies.
I love that you gave me a perfect segue, Joe. So immune system jazzed up. So a question came in as soon as you said that. What are your thoughts on the use of a CD3 versus a 4-IBB as the immune stimulating arm?
Yeah, these are discussions we have a lot. I mean, when we were going after this, obviously, when we started this program, Claudin-6 was still somewhat of a novel target, and still is. It's still early days.
Yeah, well,
And
if it wasn't novel, we wouldn't be having this.
exactly, we wouldn't be having this discussion. And so, just from our perspective, going after a novel target with its peculiarities in terms of specificity, we know that CD3 is a very well established T-cell engager arm. The other types of arms that are out there, whether it's 4-1BB, it's an activator, or for example, the NK arms, it's still early days for that. And sort of adding novelty onto novelty, I think can sometimes be dangerous. That's a more strategic type of answer. I think nobody really knows yet what's going to be the optimal sort of set of arms, and it's great that companies are exploring these different biologies, activating by, T-cell engager arms or NK arms. But I think from our perspective, CD3 is a well-established arm. And as our data shows, we get very good potent activation of the T cells with a good window between cell lysis and cytokine release. So it seemed like a good decision. It seemed like, and still seems like, the best decision for us.
We've covered a lot here today. I mean, it's very exciting news, but we kind of ran over a little bit. Thank you, everyone, for taking the time to run through these developments with us. If you have any other questions, or if you would like to speak to us one-on-one, please do not hesitate to reach out and use the contact information shared at the bottom of the press release. Have a good day.